Hello, and welcome to Plants and Python. My name is Bob Van Buren, and I'm an assistant professor here at Michigan State University in the Department of Horticulture. And so in today's lesson, we're going to be learning about the powerful Python package, Biopython, and how to work with sequences, manipulate them, and identify them using this important package. And so the learning objectives of this lesson are first to provide a broad introduction to Biopython. And so Biopython is a very large package. There are tons of different molecular biology, computational biology, and genomics applications that you can use Biopython for. Today, we're just going to scratch the surface, and then you can explore this more on your own. We're going to learn how to write, manipulate, and transform sequences as strings. We will learn how to import, filter, sort, manipulate, and write large sequence files as SeqRecord objects. This is really great for working with sequence files of various lengths. And then finally, we will learn how to compare sequences using BLAST via Biopython. And this will tie into the remainder of this lesson, where we will be doing de novo assembly on an unknown plant genome. And then you will use Biopython to manipulate the output of that, and then run BLAST and identify what species that you assembled. And so Biopython is one of many packages that you can use for bioinformatics or computational biology tools. And so this is one of the foundational ones, but keep in mind there are lots of other programs and modules in Python that you can use for all of your downstream applications. And so what is Biopython? So it's often referred to as a workhorse for manipulating and analyzing these large-scale data sets. It represents efforts from various international groups. So there are a lot of contributors to this project. And the goal is to make it as easy as possible to use Python to work with bioinformatics data by creating high-quality, reusable modules and classes. So there are a lot of different features, but some that are important to highlight here is that it can serve as a parser for various bioinformatics file formats. So things like BLAST, ClustalW, FASTA, GenBank files. This is a massive issue in bioinformatics, is converting between one file format to another or parsing it from one program to another. So that's really great here. It can provide online access to different services and databases, such as NCBI, which we'll be doing in this notebook here today. And it can interface with various common or not so common programs as well. So you can parse sequences and then interface with programs for phylogenetics and other applications. It creates a standard sequence class, which is really important, has various clustering modules, and it has a KD tree structure or tree data structure format. And so again, we're just going to touch on some of this today, but this is a really powerful package that can be used for all sorts of interesting downstream analyses. And so first, in order to run Biopython, we need to first install it. So if you haven't done it already, please do that right now. And so to do this, simply open up the Anaconda PowerShell prompt, which is what you should have used to install Jupyter the first time. And so we will use pip to do this. And so simply type into the prompt pip install Biopython and follow any directions there. It will install the latest stable updated Biopython and any dependencies that are needed. And then you might need to restart your Jupyter notebook so that it can run Biopython. And then to make sure that it was installed correctly, and we can inspect the version as follows. And so we're going to import Biopython as import bio. And to figure out the version number that you're working with, we can use the print function and then type in bio.__version__. And then if we run this, it will output this version of 1.78. And so in this case, we're using version 1.78. Your version should be the same as this, but it might be different depending on the time in which you you're doing this lesson. In general, we're using some pretty basic functions and utilities in Biopython, so the version shouldn't matter too much, but always be sure to incorporate version control into your analyses. Okay, so the first thing we want to do is introduce the kind of one of the foundational components, which is the Seq object. And so we'll discuss this in more detail later, but most of the time when we think about sequences, we have in mind a string of letters of A's, T's, C's, and G's. In this case, we're talking about DNA, but this could be DNA, it could be RNA, it could be protein. And this string here that I've produced is not a random string. It's GATTACA, GATTACA, which is a reference to the movie Gattaca. And we'll be using this, this string for this introductory material. And so you can create a Seq object with this sequence as follows. So first we want to import the Seq module from Biopython by typing in from bio.seq, import Seq. And then we're going to create our variable my_Seq by using this Seq function, and then create this Seq object, this string of letters here, Gattaca, Gattaca. So just type that in. And then if we call our variable here, it will return the Seq object, and then our sequence here of Gattaca, Gattaca. So this is a string, so we can also simply use the print function to print our string. And then this will print our sequence of DNA as well. So this is essentially a string, but it's not like a normal string. So in Biopython, we can do things with our string of DNA, RNA, or protein that you can't do with normal strings. And so we could, let's here start with print again. And so again, this is the sequence that we're working with. And then we can use the function called complement. And in this case, we're going to take our sequence string here, and we're going to provide the nucleotides on the opposite strand of DNA. And so we know that G's are always opposite to C's, and that A's are always opposite to T's. So if we complement it, we're going to get the complement, the opposite strand, and then here we're going to output a sequence object where basically the G's are complementary to the C's and the A's are complementary to the T's. And so this is really great. Maybe you want to transform it to do some molecular biology stuff. Maybe you need to reverse complement it to get the opposite strand, because sometimes transcripts run in one direction on the DNA, sometimes they run in other directions. And so we can do that as well. By instead of complementing it, we can reverse complement it, where we go on the opposite strand and then we go in the opposite orientation. And so we can use this function reverse complement here. And again, we're just going to run this on our variable mySeq. And then if we type that in, this is our original sequence here. And then we have the reverse complement where we've taken the sequence on the other strand of the DNA and then we flopped it around. And so we could use this to do protein prediction. You can do this for primers. So when you're designing primers for PCR, you want one primer to be on one strand of the DNA going in this direction, and then you want the other primer going in the opposite direction so that they will meet up and you can amplify a fragment of DNA. And there are other things that we can do as well. So you can't really do this with a normal string. So in many ways, we can deal with them as strings, normal strings. So we can do things like we can get their length, we can iterate over the elements, we can get specific elements of the string, or we can add strings together as well. And so we're also going to import this module codon table. So this will allow us to translate DNA into protein. So let's do that really quickly. And so we can access elements of the sequence in the same way as for normal strings. And so again, if we have our sequence object here, mySeq, again, it's going to have the sequence Gattaca, Gattaca. So we can simply, we can print the first character here, which is going to, remember Python always starts with zeros. So we could print G. We could print the third character, which would be a T, or we can print a segment of our string here. So the third character which would be a T, all the way to the 10th character, which would be, I believe, this first T here. Yes, and so we can treat it like a normal string where we can manipulate it. We can take bits and pieces, we can chop it up, we can put them together. We can also add two strings together. And so we could create two sequence files. In this case, we will call them mySeq and yourSeq. So mySeq will have, the sequence object will have the sequence of Gattaca. The yourSeq object will have the basic, the reverse of Gattaca. So ACATTAG. And we can simply use arithmetic and add myString to yours, or mySeq to yourSeq. And then we get this sequence object that is basically these two objects concatenated on top of each other. So you can add them together, you can have a whole bunch of sequences, add them together, and it will just concatenate them into one singular seq object. So we can do a lot of the things that we can do with a typical string as well. Okay, we can also translate our sequences of DNA or RNA to protein. And so again, this is something that you can't do with a typical string. And so let's print mySeq. And so in this case, it's just Gattaca. And then let's translate this. And if you remember in molecular biology, each 3 letters of a DNA or RNA molecule correspond to a codon, which specifies one of 20 amino acids. And so we could create a dictionary and keys, and we could do this, or we can use this simple function .translate. And so what it's doing is it's looking at the 3-letter combinations, and then it's outputting the amino acid that corresponds to that. And so in this case, mySeq is a very short sequence. This would be a short peptide of only 2 amino acids. And we also get this error message here that we have a partial codon because the length of the sequence is not a multiple of 3. And so in this case, we have 7 letters. So this first one corresponds to this amino acid. These TAC corresponds to this amino acid, and then it encounters an A. And since that's not a codon of 3, it throws this error message where it's a partial codon. So we can use this to translate our sequences. And so maybe we have a couple hundred or a couple thousand individual sequences. We can use this translate function to quickly turn those from nucleotides into proteins. So some sequences translate well, where it's just a string of proteins, but then you can also introduce stop codons as well. And so here's a random sequence that I've called my bad seq. And so it's just a random strings of A's, T's, and C's and G's that are randomly typed into the computer. And then if we try to translate this sequence, we get this random character in the middle here. And what this specifies is a premature stop codon. And so as we were translating each of these 3 codons, it encountered a TAG, I believe, which is the typical stop codon. And so if this were a ribosome, as the RNA molecule were moving through the ribosome, if it encountered a stop codon, it would just release the peptide, release the piece of RNA, and stop. And so it's really important that if you're manipulating sequences, if you're creating your own sequences, that you look for these premature stop codons. So far we've learned how to work with individual seq objects. These are individual strings that might have a couple hundred to a couple thousand characters. But in general, when we're doing bioinformatics, genomics, or computational biology type analyses, we're working with large files. So you might be working with a genome that could have dozens to hundreds of chromosomes or contigs in it, or you could be working with a raw sequence file, such as a FASTQ file from maybe Illumina or PacBio sequencing, where you might have millions or billions of individual sequences. And so when we want to work with more than one sequence at a time, we can use this powerful function, seqio.parse. And what this will do is it will read in sequence data from an external file, and it will store them as SeqRecord objects. And so to use this function, we need to supply 2 different pieces of data. So it requires 2 arguments. So the first argument is a handle to read the data from, or a file name. And so in this case, it would be an external file that we would provide. It could be something if you're running this on a high performance computer cluster. It could be a local file there, it could be something on your computer, or it could be a file downloaded from the internet. And the second argument is a lowercase string specifying the sequencing format. And so Biopython won't try to guess the file format for you, so we need to provide this. So there are a couple different cases that we could have here, but in our course, we'll mostly be talking about the .fasta and .fastq formats. And so if you remember, the .fasta format is going to be a list of sequences where each one is separated by a caret or a greater than sign with a name for the record. And the .fastq is somewhat similar. We'll get into this in the next lesson, but this is the file format that you would typically see for large-scale Illumina, PacBio, or nanopore data, where it provides the sequence and also some sense of the quality of the sequences as well. And so if we supply these two things, we can import data files of virtually any size. Okay, and so SeqIO parse function returns an iterator, which gives the SeqRecord objects. And so we typically, in order to call all the records in a large file, we would need to use a loop function, and we would run a loop to run through each record in the file. And so this is really important for the parse function is really good for you for working with anywhere from dozens to millions or billions of files. But sometimes you'll have a file with a single record. And so this could be maybe you're working with a single chromosome, or perhaps a bacterial genome with one chromosome or one contag, maybe working with a plasmid or something. And so if you're only working with one SeqRecord, you can use this other function, SeqIO.read, which takes the same arguments as .parse, but it only works with one record. So if you're working with one chromosome or just one sequence record, for simplicity, you can use the .read function, but typically in this course and typically in a lot of applications, you'll need the .parse function. And so we're going to be using in this example, this file here, this sequences.txt. So this is a file that I provided for you. You can find it on Discord or the Plants and Python GitHub page or the Jupyter book. And so when you're running this, make sure that this file has the same name and is in the same path of your notebook. So either you can have it in the same path or you can provide the full path on your local machine. Okay, and so in order to run SeqIO.parse, we first need to import from Bio the module SeqIO, and then we will create our file here, seq_file, and then we will run seq.io or SeqIO.parse function, and then provide it the two inputs that we need. So this is the first one is going to be our sequence file. And so again, specify either the full path, or in my case, this file, I have moved it into the same path as the notebook on my local computer. And then we also need to tell it the sequence type. And so in this case, it's a FASTA file. So if we run this, we're not actually going to get any output because it's just basically parsing this data set. And so it's kind of storing all the records in there. So it's not actually running a whole lot, but we can use a for loop to print out basically what we have in this file. So let's create a for loop. So we will say for record in, and we'll call our seq file. And then we will say example equals record. And so in this case, we will iterate over each record in the file. And so this is each individual sequence in our file. And then here, since I just wanted to go through one time and output the first record in the file, we'll just use break. And so in this loop, once it encounters this break function, it will essentially stop. And so if we wanted to iterate over the entire file, we could include another argument here and not include break. But in this case, I just want to print basically the first identifier. And so then we will do print, and we'll print record, and we'll run that. And so now we have basically, we have iterated over the first record in our file. So this is the first sequence, and then it's output some features here. And so the name of our first sequence is OT underscore blah, blah, blah. And so what this means is this is a standard way to denote genes in a plant genome. And so OT is the species. So in the last part of this exercise, we'll talk about what the species is. This is the chromosome number. So this gene is on chromosome 10. And then this is the gene name. So the ID, the name, and the description. In this case, I've only provided one of these, so it's going to give them all the same name. And then it's going to output our Seq object here, where it's going to show the start of the Seq object and the end of the Seq object. And so again, this is a gene. So it's starting with ATG, which is the standard start codon, and then it is finishing with the TGA, which is a stop codon. And it will not output the entire sequence by default, just kind of a snippet. And so this is what the first record in our file is. And then maybe we want to see all of the records in our file. So in this case, we will do something a little bit different. So we will use, again, a for loop. And so we use the same for loop here, a for record in our Seq file. We will print the record.id. And so this is going to print, if you remember from the previous cell, it will print the sequence header. We will print a representative piece of our sequence fragment. So this is what this command here is. And then we can print the length of each sequence in the file. And so what this for loop will do is for each record, so for each sequence in our file, it will print the ID, it will print a representative chunk of the sequence, and then it will print the total length of the sequence. And then it will iterate over every entry in our file. And so if we run this, we basically get 1, 2, 3, 4, 5, 6 sequences here. And we have, again, the name. And so these are single genes, but this could be a chromosome. It could be just some random piece of DNA. This is the sequence, what it starts with, what it ends with. And again, you'll notice that these are typical stop codons. And then this is the length of the gene here. And so these are of different lengths. This is just a snippet of a couple genes from the genome, but you could use this to read in a file of basically any size. So you could read in a genome, you could read in raw sequence files, whatever you want. Okay, so we can also treat these sequence files as dictionaries. And so let's say we imported a massive sequence file that had millions of entries, but you only wanted to export maybe a couple dozen of those. And so what we can do is we can create a Python dictionary using this sequence file. And then we can use this to access certain elements of the file or a subset of sequences. And so we can create dictionary and keys, and then we can call individual keys and export them to a separate file. And so, a big challenge in bioinformatics generally is working with different data types and parsing files, removing things, cleaning them up. And so this is a really great way of taking a subset of a file using identifiers. And so the function that we'll use to do this is seqio.to_dict. And what this will do is it will make a SeqRecord dictionary. And then this will use the identifier, the ID property of each sequence as the key. And then we can export this, or we can write out a different sequence file. And so to run this, we will first parse our sequence file again. And so again, we have Seq file. We're using this parse function here, providing our sequence, and we're telling it the FASTA type, or the type, which is FASTA. And then we're going to create this dictionary here. So we'll call our dictionary grass_dict. And what this is some sequences from a grass genome, as we'll see in the last activity. And then again, we will do seqio.toDict, and then use the Seq file. And so what this is doing is it is creating this dictionary here. And so we've created our dictionary now. So let's look at what some of the keys are, some of the attributes of our dictionary. So then we can print, let's start with the length. So we'll see the length of our dictionary. And so we'll use the len function and then provide our dictionary. So grass_dict. And so our dictionary has a length of 7. So there are 7 keys here. And we can also print the actual keys themselves. So let's use again the print function. So grass keys. And then this is going to print the actual keys in here. And so these are, again, our identifiers. So these are the same ones that we saw before. And so if we want to call individual elements from this dictionary, we can just simply use these gene names here. So again, in this case, we're going to show a relatively small file, but you can use this for files of basically any size. So let's say we want to access a single entry from this dictionary. And so what we would do is let's create this new file here, this SeqRecord. And so we will access our dictionary here, and we will call this single gene here. And then we'll create this new variable here, and then we can look at some of the attributes of this. So we can print the description, so that's going to be the ID. We can print, again, representative chunk of the sequence, and we can print the length. And so if we run this, we've created this new SeqRecord here, which is a Seq object. And it is, instead of having the file with all 7 sequences, it's created a file with a single sequence from our dictionary. So again, this is a really powerful way of like separating out individual components or subset of sequences from a much larger input file. So right now we've just stored these as SeqRecord objects, but maybe we want to export them into a sequence file. And so we can use this function seqio.write, which is pretty self-explanatory, which will write sequence outputs. And so instead of storing it as a SeqRecord or Seq object in our notebook, we can write it out to whatever file format we want. And so the write function takes 3 arguments. So we need some SeqRecord objects. So we've created these already. So this is going to be like our subset of sequences that we want, a handle or file name to write to. And so this is whatever the file we want to call it. And by default, it's going to write to whatever the local path is of the notebook. You can also specify a full path on your local computer, or if you're working on a high performance computer, you can provide the full path there. And also the sequence format. And so it's very similar to the parse function, except in addition, we're providing it basically our list of Seq files, and it will output this file that we create. And so again, we're going to run, we're going to go into our grass dictionary here. We're going to create this SeqRecord here of this single grass gene. And so in this case, it's OT_chromosome 10, blah, blah, blah. And then we're going to use the .write function. And so seqio.write. And again, we need to provide 3 arguments. So we need to tell it the SeqRecord object. And so that's this single grass gene here. This is the file name that we want to output to. And so we're just going to call this grass_gene.fa. And .fa is a typical file extension for a FASTA format. And then we're going to export it as a FASTA format. And so if we write this, this has created a file here of grass_gene.fa. And if you pause this video and you look in your local directory that has the Jupyter notebook, you will see this new FASTA file here. Okay, so that's kind of where we're importing things. We can write them, we can take a subset. Maybe we want to do something like sort them by different character attributes or sort them by length. And so this is really important for working with large raw input FASTA files. Sometimes they need to be sorted before a program can process them. And so we can use this sort function here. And so this will sort by, it can sort by characters, it can sort by the length, it can sort by all kinds of things. And so first, let's sort these sequences by length. So if you remember before, they were just kind of randomly put in the file, but maybe we want the shortest sequences first, and then the longest ones at the end. So the first thing we want to do is parse our sequence.txt file here, and then we'll create this Seq object records here. And then to use the sort function, so we will do records.sort, and then we'll use this key equals lambda, which is, you run that in a function where you have kind of an unknown or wild traits that it needs to run through. And then what this is going to do is sort everything in here by length. And then we can write our new file here. So this seq object will be sorted by length, and then we can output that by specifying our seq object, the file that we want to call it. So we're going to call this one sequences_sorted, and then the sequence type. Okay, and so this is just basic outputting it, but again, we don't know all the character traits here, so we can write a simple for loop, and we can parse this file again and then see what the traits are. And so I'm just going to paste a for loop into here. So very similar to the previous cells. So in this case, we're going to write this for loop. We're going to parse this sequence file that we just created, sequences_sorted.txt. And then we're going to print for each record in the list, we're going to print the sequence header and the length of each sequence. And so now, instead of having these sorted randomly by just their name or whatever random trait we had when we imported the file, these individual genes are going to be sorted by their length. So the shortest gene is 522 base pairs, and the longest is 3,357. So this is really helpful if we have kind of a raw input and we want to sort it by any set of characters. So, and then so we ran this already, so let's make sure that they're sorted. Okay. And let's say we wanted to sort it in the opposite direction. So instead of sorting it smallest to largest, let's say we want to sort it largest to smallest. And so we will do essentially the same thing, but instead of sorting it by length here, where it's going to go smallest to largest, we can put this negative sign here, and then it will sort it by largest to smallest. And then we will create, so we will create the seq object here, which will basically inverse the orientation. And then we can write that object by providing the seq object name here, the name of our file that we want to create. In this case, we're going to call this one sorted reverse, and then specify that it's a FACET type. And then we can import that new file and make sure that they're sorted correctly by using this, again, this for loop. And so if we write this, if we look at the output here, we can see that it's created this new sorted underscore reversed file where instead of them smallest to largest, these sequences are largest to smallest. And so again, this is just showing like different ways that we can sort text files. So the last thing we're going to do in this activity is identify the origin of the sequences that we've been working with. And so I provided the sequence.txt file to you. I've told you that it's a grass, but maybe we want to figure out the exact species that it came from. And so we can use this powerful algorithm BLAST. BLAST can be used to compare sequences against each other, or it can be used to compare sequences against a database. So BLAST can be used to identify homology between proteins, DNA or RNA molecules. And so the BLAST database in NCBI has, at this point, probably billions or trillions of entries from tens of thousands or even millions of different species. So fungi, bacterial, plant, humans, mammals, take your pick, it's all in there. And so maybe we are sequencing a pathogen and we get some sequences, but we don't know what that pathogen is. Maybe we have some contamination in one of our samples and we have sequences of it, we don't know what it is. So what we can do is take a sequence of unknown origin and we can compare it against all of the other sequences that have been compiled over the last 30 or 40 years and identify what it likely came from. We can also use it for comparative genomics. So we could take genes from one genome and genes from another genome, compare them all to each other, and then see which genes are orthologous to each other. So that's what we're going to be doing in the next notebook. And there are a bunch of other things that we can do here. So BLAST is kind of a foundational tool in molecular biology and genomics. And we can actually run it locally here over the internet using Biopython. And so this is kind of an example of what a BLAST output might look like. And so here we have our query. So this is the sequence that we're aligning everything against. And then, so this is 3,000, probably 300 base pairs or so. And then this is the output, or the sequences that align to it. And so some of them are aligning to the whole length, and some are just aligning partially. And when we actually run this, we'll talk about what the output looks like and how we can utilize it. So when we run BLAST in Biopython, we're going to be running it over the internet. So you can run BLAST locally on your machine if you have a database of sequences that you want to run it against. You can run it on a high-performance computing cluster, or we can run it over the internet onto NCBI's massive database, where there are, again, probably billions of sequences there of known origin, and we can see what our sequences are most similar to. And so to run this function, qblast, we need to import the bioblast NCBI www module, and this will call the online version of BLAST. And so to run this function, we need to provide 3 non-optional arguments, and there are a bunch of other optional arguments that we won't really talk about today. So the first one is a BLAST program to use for the search as a lowercase string. And so there are different, BLAST can work with different types of sequences. So it can work with DNA sequences, and so this would be BLAST-N. It can work with protein sequences, which would be BLAST-P. It can work with basically taking DNA molecules or RNA molecules and translating them into proteins, and then searching against a database of proteins. This would be like BLAST-X or the inverse of that, and which is T-BLAST, or both, where you take 2 DNA molecules, you convert them into proteins in each of the 6 possible reading frames, and then compare to each other, and that's T-BLAST-X. So we need to specify one of these. And in our case, we could use BLAST-N because we have genes, or we could use the program BLAST-X, and we can take our gene sequences, convert them into proteins, and then compare them to all the proteins in NCBI. The second argument we need to specify is the database to search against. And so there are lots of options for this. We could use a local database that we've compiled on our computer, or we can use the massive databases that NCBI has generated. And so if you're trying to compare one sequence against a sequence of maybe genes from a species, a closely related species, or if you're trying to compare 2 genomes against each other, we might want to provide a local database that we create. But if we don't know what our sequence is, we might want to blast it against everything that's out there and have the highest chance of returning a full hit. And so we can specify that. And then the third argument is a string containing your query sequence. And so this can be the sequence itself. It can be the sequence in FASTA format as a SeqRecord object, which is what we'll be doing. Or you can even provide an identifier like a GI number, and this would be an identifier that would be on NCBI. And there are a number of other options that you can run here. So things related to how to output the files, different parameters using for filtering or for clustering or alignments, overlaps, that type of thing. But for simplicity, we won't get into those today. Okay, so the first thing we need to do is from bio.blast, we need to import this module NCBI www. And because this is sort of a complex thing and there are lots of arguments that we can feed, we can use the help here and output all of the possible arguments that we can provide. And so if we type in help, we will get this useful output here. And so this is just providing some all the different arguments that we can provide here. So it talks about the program, talks about parameters, and then these are some of the different parameters that we can set. So we can set the program, which is what we're going to be doing. These are the 3 that we need to have. So there's a BLAST-N, BLAST-P, etc. The database to use, the sequence to search, and then these are just a subset of the other optional things that we can output. And so, for instance, you can change the number of alignments to show. So the default is 500, but we could show 10, we could show 1,000. We can change the e-value cutoff, which is basically a statistical test of how likely it is that your sequence matches another sequence in the database. We can turn on filtering, we can change the format type, et cetera, et cetera. So there's lots of things that we can change, but in this case, we're just going to do a couple things. And this is something good to remember for a lot of programs you'll run with bioinformatics is there are lots of things that you can change and tweak depending on the application that you're running. Okay, so let's try blasting the previous file we wrote. So this is the grass.gene.fa. And so if you'll remember, this is a sequence file with a single grass gene in it. And let's see if we can figure out what species this is. And so we will use the seqio.read to read our FASTA file. And then we will use this .qblast to blast it. And then we will write that file and then take a look at it. Okay, and so again, we're going to call from bio.blast, we're going to import ncbi www, and www stands for basically that this is going over the internet. And then we're going to create this query here. And so we will use seqio.read to read in the FASTA file that we created. And again, this FASTA file has the format FASTA. And so this will create our query file here, our query stored SeqRecord. And this will just have basically a single gene in it. And that's going to be our grassgene.fa. And so if you remember, we can use this .read function instead of .parse. And this will be useful for reading in a file with a single entry. And then we will create our output here, which is going to be our research handle. And then we will run our function ncbi.www.qblast. And then again, we need to provide it at a minimum of 3 arguments. So the first is the program to run. And so since we are looking at DNA molecules, we will run the program blastn. But if you're working with proteins, it would be blastp, or there are a couple other options as well. The database that we're going to be using is, this is NT, which is nucleotide. And so this is basically everything on NCBI. So there are a number of different databases that you can use. You can use ones that's just the human genome. You can specify any genome that you want. You can do a subset of just plants or just bacteria or just viruses. In this case, we in theory don't know what this sequence is, so we're going to go against everything. And then we're going to run it against this file that we've created, this query file. And then we're also going to output the format type as text. And that's so that we can read it more easily in the Jupyter Notebook. And so if we run this, this is going to take a little bit of while to run, so it might take a couple minutes. And so what it's doing right now is it's created this, this SeqRecord here with the single grass gene in it. It's taking that SeqRecord. It is, we have to be connected to the internet to run this because we're doing it over the internet. And so it sent this to NCBI, and it's currently blasting it using the algorithm against all of the sequences on NCBI. So it's going against billions of different sequences and trying to find which ones have the highest homology. And so if you're running like a single gene like we are, this could take 30 seconds to maybe 5 minutes or so. If you were to run an entire chromosome, it could take days or weeks. And there are better programs that you can use for larger scale, things like that. Or you could give it, instead of a single gene, you can give it like, you can parse a file with like hundreds or thousands of genes and run it against here. But just remember, the more genes you provide it, the more sequences, the larger the sequences, the more time it will take. And so that took about a minute, and let's see what it output. Okay, so when we're doing this, we have to be a bit careful because we've output this temporary file, and we can only use result_handle.read one time to read the BLAST output. And so if you call it a second time, it will return an empty string. And so if after we run this next function, this read function, the results are in a file that we'll create here, and then the original handle has lost all of its data since we closed it. So when you're basically reading this BLAST output and then converting it to a text file, make sure you only run this once, because if you run it a second time, that string will be empty. So just be a little bit careful when doing this. And so we will use this command with open, and we will open our mytext.blast. So this is the output BLAST file that, or this is the output that we want to create here. And then with open as, we're going to save it to this format. So we're going to use our command save to.write. And then we will use our result underscore handle.read. And then we will close this out. And so what this will do is it will read in the temporary BLAST output that we've created. It will save it, and then it will basically write it to this file here, my blast.txt. So if we run that, again, we're not going to see anything, and then we can see what that file had. And so now that we have successfully saved our output, again, we can use a loop to read the file and identify what species this came from. So created my file here, so we're going to open our output here. And so this will be saved again to whatever directory you're currently in for your notebook. And so you can open this with a text viewer as well. You can also specify any directory in your local machine or a computing cluster as well. And so we'll use this, basically this for loop, so that we can print every line and scroll through it. And so we will make a for loop of for line in my file. So this is going to be every line in the file. It will basically print that line, and then it will iterate over every line of the file and print it as an output. And then we will close that file. And so if we run this, it's basically going to be printing it line by line, and then we can scroll through and see what we have here. And so just looking through some of the outputs here. So this is the version of BLAST that it ran, 12.2. This is like if we needed to cite this paper. We ran against this nucleotide database here, this collection. And so this collection has 73 million sequences, which correspond to 519 billion total base pairs. So that's a ton of sequences. And so it ran only a minute or two, so that's pretty impressive. And so our query sequence itself had a length of 3,357 base pairs. This was a gene. And then we can see what the significant alignments were. And so if we look at this output here, the first thing we're going to have is some random identifier, NCBI. We don't care about that. And then this is going to be the species name and the gene that it aligns to. This is a score. And then the things that we care about here are the e-value. And this is a statistic of basically by random chance how likely this is to match our sequence that we've provided, and a maximum identity. So this is how similar the sequences are on a nucleotide level. And so in this case, e-value of 1 would mean like it could just be something random by chance, and e-value of 0 means that it is almost certainly corresponding to the sequence. They do have homology. And then basically anything with an e-value of less than 1 times 10 to the 1 to the negative 10 is considered a pretty good e-value. And so if we look at these things, we have a couple different grasses here. So Phragmites is kind of a weed that you would see around. We have switchgrass here, which is an important biofuel crop. Soteria, which is an important millet and model species. Sugarcane, sorghum, rice, etc. So this is definitely a grass, but you'll notice that none of these have a max identity of 100%. And that's because the grass that we're working with here is not in this database. And so the first thing we'll output is basically almost like a table here that talks about what the hits are and their identity. And then if we scroll down a little bit, we can actually look at the alignments themselves. And so on the top, you're always going to have your query. So this is the sequence that we provided. And at the bottom is the subject or the sequence from the BLAST database that it aligned it to. And so if those sequences match, we will see basically a line that connects them. And then if there's a difference, we will see a missing line here. So you can see in this position, our query is a T and the subject is a G. And so here, this identity is 92%, which is actually pretty similar for different grasses. And it's pretty high quality. So we can scroll through here. By default, it will output 500 individual sequences, which is probably what it output here. So you can see this is a massive file. We might only care about the first couple entries, and you can specify that as well. So we could use this to identify the species that we're working with. In this case, our species is Orthopedium thomium. So it's a grass that I work on in my lab. It's not in this NCBI database, but we can see some of the other species that it's closely related to, and we can also see what the gene is. And so in this case, the first gene, or this gene, is likely a plasma membrane transporter. So that's what these genes have been annotated as. And so maybe if this were a gene we identified as important in our trait of interest, we could then look at these genes in more detail. So BLAST is very powerful. Again, we've just kind of skimmed the surface of what we can do in Biopython. But in the next activity, we're going to be blasting whole genomes against each other and then doing some comparative genomics. So just to summarize what we learned about today. So we provided a broad introduction to Biopython. And so remember, Biopython is a really powerful package. So we've just kind of skimmed the surface of what we can do here. If you want to read more about it, I would recommend going to just Googling Biopython. You can go to the page and see all the different things that you can do with it. And this same goes for tons of different modules in Python. There are a couple that we'll use in this course, but there are literally thousands, and they can do all sorts of powerful statistics, machine learning, image analysis, and various genomics, bioinformatics type applications. So a lot of really powerful packages. If you have time and you're interested, take a look at that. So with Biopython, we learned how to write, manipulate, and transform sequences as strings. We learned how to import, filter, sort, manipulate, and write larger sequence files as SeqRecord objects. And so this is, in this case, we use it for a file with 7 entries, but you could use this for millions or billions of entries, so really powerful. And then we learned how to compare sequences using BLAST via Biopython. And so you can run BLAST in all sorts of different formats, but we just showed it using Biopython. And so for the rest of the activity here, you're going to be assembling a reference genome in class. And so I'm going to give you a series of unknown reference genomes, and then you can use Biopython with the output of that to sort, manipulate that output, and then utilize BLAST to tell me the identity of the genome that you sequenced.